How do you record? Start recording. Oh, it is being recorded already. Fantastic. Okay, yeah, don't mind it being recorded. That's fine. Right, so here we go. So like I said, so we're going to do the sulci, the gyri, then the median sagittal view. We're going to look at this idea of the homunculus and apply it to the cortical layers of the brain. This will all make sense later, don't worry. And then we're going to grossly, and I mean really grossly, look at the blood supply of the central nervous system. So for those of you who don't know me, my name's Dan. I am an actual anatomist. I taught at Liverpool and Manchester Medical School before coming here to be a medical student for some reason. I don't know why I thought this, but you know, here I am, why not? So, David Tennant, I want you to look at a skull the way David Tennant looks at a skull. These are the different bits of the skull that you need to know as a medical student, particularly for phase 1. You need to be able to point to all of these different bits on a skull. So we're going to go through them. So if I bring up a 3D skull here, I'm going to talk you through these things. So hopefully this will work. So we'll begin with the frontal bone. This is the frontal bone at the forehead. And the parietal bone, if we pull it over to the side, parietal meaning wall, because it's the main substance of the wall of the calvarium, which is the posh term for a brain case, the top of the head. So this is the parietal bone. Then we have the temporal bone. Temporal, meaning like if you think about temporal and spatial summation and action potentials, temporal means time, because these bits of hair are the first bits to go grey, where the temporal bone is. Occipital bone literally means back of the head. And that's this region here. So this is your occipital bone. And then if we whip the skull back round to the anterior, you can see within the orbit this bone called the ethmoid bone. So this is the ethmoid bone right here. Then right at the front of the nose, where a lot of lay people squeeze their nose when they're having a nosebleed, is the nasal bone. Obviously, you should squeeze the soft part and lean forward instead of back, because if you lean back, then you swallow the blood, so forward, soft part. So these are the nasal bones, paired structures. The lacrimal bone contains a duct into which your tears go into the nasal cavity. So you can find it just here. Here's your lacrimal bone, and the lacrimal duct is just sort of here. The maxilla, quite common sense, the maxilla contains the top bit of teeth. We call that the superior arcade of teeth. So here's our maxilla. Our zygoma means yolk, like the yolk of an egg. Don't ask me why. But this is the zygomatic bone. And the zygomatic bone articulates or joins with a couple of different bones. So the frontal bone up here, the maxillary bone here, and the temporal bone here. So this is our temporal bone. And this is the portion of the temporal bone called the squamous temporal bone, which means a flat bone. So it has a little process that abuts out here and then runs anteriorly to articulate with the zygomatic bone. And this forms, both temporal and zygomatic bone, this forms what we call the zygomatic arch. And the zygomatic arch is the origin of the muscle called the masseter muscle, which originates from here and goes right down here to attach at the angle of the mandible. And it will contract from here to there. So it closes your jaw very forcefully. They're the powerful chewing muscles. You can sort of chew and then you can feel your your masseter muscles like so. So sutures now. So in terms of the sutures, and I'll just mute my phone, sorry. In terms of the sutures, there's not many we can see from here. What we can briefly mention at the back here is the lambdoid suture. This sort of like upside down Y shape, or the Greek lambda symbol, is here. And that is the union between the single occipital bone and the two parietal bones here. This is the lambdoid suture. And then this wiggly thing here is the sagittal suture, as in Sagittarius with an arrow in mythology. It shoots straight ahead, so it's a straight line. Well, it looked wiggledy, but it's going straight forward anteriorly. So that's Sagittarius. And then if we pull around to the side here, what we can see is the coronal suture. And the coronal suture sort of runs along the top of your head like this. And the coronal suture is named for a crown of garlands, because the old Roman crown of garlands was like flowers that sort of like splayed out like this. So you have sagittal going that way, you have coronal going that way. And then the other thing to talk about is the fontanelles and the orbit. So we need to look at from the top here. This is a supra-inferior view to talk about the fontanelles. Now, if you've seen a neonate, if you've seen a newborn baby, you'll note that there are holes in the skull at the top. There's an anterior fontanelle and a posterior fontanelle and a few others along the side that we don't need to worry about for now. These are formed because when you're born, your brain is soaking in information. It's all novel. It's pretty boring inside a uterus. And then you're born, and then things are happening everywhere. The sounds, there's God knows what, right? And so your brain's absorbing all this information, and it grows so rapidly that your skull cannot grow quick enough. So what the body does is it says, hey, I need to keep some gaps in between these bones so there's space for the brain to grow. Because if you're growing outwards and you hit bone, then it's going to get squished, you'll get raised intracranial pressure, it's really bad. So any sort of ossified fontanelle or fontanelle that hasn't formed properly is a serious problem for neonatal development. So this is just fibrocartilage that holds these bones together. And over the course of a couple of months to a couple of years, these solidify. So the anterior fontanelle becomes what we call the bregma, which is the union of the coronal suture and the sagittal suture. And then the posterior fontanelle at the back here becomes the lambda, the union of the lambdoid suture and the sagittal suture. So those are a few different structures for you to know there. And then finally, we'll just briefly touch on the orbit. Now, just have a rough idea of these bones. It's not so important because you're going to do orbital and eye anatomy in phase 2, but I can't really talk about the skull without mentioning them. So obviously blue here, this is our nasal bone, this is our maxilla right here, this sort of dark purpley color. This is our zygomatic bone, which we said is sort of like fanning out and articulating with the temporal bone here, as well as the frontal bone up there and the maxilla along here. This is the lacrimal bone, and then we have the ethmoid bone, and then we have a bone here which sits in the middle of the skull and looks like a butterfly, and it's called the sphenoid bone, and we'll meet that a bit later. And then here, this is the frontal bone, obviously. So, orbit, just have a rough idea. In terms of examinable things for phase 1, not so much. But what is examinable for phase 1 are the cranial fossas. So you take the top of the skull off. So what we've done, and what you actually do when you're doing dissections, which sadly I had to do for about 6 years, is you take a saw and you saw around the frontal bone and the parietal bone, and you get a bit of the occipital bone here, and then you lift it off, and then you open up the dura, you pull out the brain and dissect away the brainstem, and you're left with this view here. So we're looking at the different bits of the base of the skull. So the brain will sit in here. So we divide this complex mesh of bone into 3 different areas. There's the anterior, the middle, and the posterior cranial fossa. And just remember that fossa means a sort of depression, a groove, a cave, if you will. So the anterior cranial fossa, for the most part, is formed of the frontal bone, a little bit of the ethmoid bone, and the lesser wings of the sphenoid up here. So mostly you can think of it as frontal. And the 2 things that you need to know for phase 1 is the Crista galli, which means the cock's comb, you know, sort of like a cock, like the rooster crowing, like this. It has sort of like a fan thing on the top of its head. That is what this central structure is here, and it sticks out, and it's where the dura mater attaches to form the falx cerebri, which we'll talk about a bit later. So the dura mater attaches to the Crista galli here. And then there's perforations here in the ethmoid bone called the cribriform plate. And that's where the fibers of cranial nerve 1 come from the nose up into the brain case. So that's all you need to know for the anterior cranial fossa. The middle cranial fossa is a bit more complicated. It's mostly the sphenoid bone, but there's a little bit of temporal bone, a little bit of parietal bone as well, thrown in for good measure at the back here. And there's lots of holes you need to know. So lots of holes. We're going to deal with the holes in turn, but what I want to draw your attention to right now is this central structure here. This is the sella turcica, which means saddle that is Turkish, because the pituitary gland sits in this sella turcica. It comes from the hypothalamic region in the base of the brain and projects down. So a stalk hangs the pituitary gland like a conker on a string into this hole here. And there's various different names for all these different protrusions of bones, which are called the clinoid processes or the clinoid processes. You don't need to know that. What you need to know is this is the sella turcica. And the reason you need to know this is if there's any sort of pituitary adenoma or any kind of pathology like that to do with the pituitary gland and a neurosurgeon has to take it out, one of the approaches is called the transsphenoidal approach, because this is the sphenoid bone. So they go up through the nose and they break the back of the nasal cavity with a little hole. They drill in, and that opens up into the Turkish saddle here, so they can access in as littly, well, I can't really call it non-invasive, but you're not sawing the skull off, in as limited an invasion as possible to get to the pituitary gland. So that's what you need to know for that, and we'll come to the holes in a minute. Now, the posterior cranial fossa is pretty much the same. Now, these are all holes apart from this word called clevus. Now, clevus is this ramp here. So you have the foramen magnum, literally hole that is big or hole that is greatest, the foramen magnum, and then this ramp up here towards the pituitary gland. And this ramp is where the pons of the brainstem sits. And on the pons is a giant artery called the basilar artery. And so you have pons, basilar artery, and then clevus. So they all sort of sit forward on this bony structure, this bony ramp. So it's an important bit of bone to know. So I've done this is from the Complete Anatomy app, and I've sort of color coded in the different bits of anatomy that you need to know. So we're looking from the back of the skull, and we've taken off the parietal bones, and we're looking in. So here's the crista galli, and here is the cribriform plate of the ethmoid bone. We're going to look at the holes now, these foramina of the middle cranial fossa. So we're going to talk about three here for starters. So the red one is the optic canal, and that transmits very imaginatively the optic nerve, cranial nerve 2. The second one is this giant one called the superior orbital fissure, or the SOF. And this transmits a lot of different nerves. It contributes to 4 of 12 cranial nerves. So if you're thinking about a single hole in the skull that Tunstall or Erin might write an exam question for, the superior orbital fissure, yeah, kind of. So the cavernous sinus sits here, and that's where the internal carotid artery is, and a venous plexus is, and all of these cranial nerves, and they run out into the superior orbital fissure. Yeah, so you need to know the superior orbital fissure. You just need to know these structures. 3, 4, 6, and V1. It's also called VA. It's the ophthalmic nerve, or the ophthalmic branch of the trigeminal nerve. Now, 3, 4, and 6 are your cranial nerves involved in extraocular movement, and I believe you're having a lecture on cranial nerves soon, so I won't go into more detail on that, but it's into the superior orbital fissure, and it's the cranial nerves that move the eye, and one that is sensation to the forehead, the head up to the tip of the frontal bone up into the tip, the highest point called the vertex, the highest point of the skull, as well as, importantly, the sensory supply of your blink reflex. So you take a cotton wool wisp and you go for the lateral portion of the eye called the lateral canthus, and you just wisp it with some cotton wool and then you blink. And if you blink reflexively, that's because V1 has transmitted that information through the cornea into the superior orbital fissure and then into the pons. And if it's done that successfully, then, you know, that's a test of neurological function. So we've done optic canal, superior orbital fissure, and now we're doing this one. And this one's called the foramen rotundum. And this transmits V2, or VB it's also called. And VB is the maxillary branch of the trigeminal nerve. So optic canal, superior orbital fissure, foramen rotundum. Now, I've turned the skull slightly so we can still see the optic canal, we can still see the superior orbital fissure, and we can still see the foramen rotundum. So we'll do another couple here. So this one here is actually not that well animated by the app. This is called the foramen lacerum. It's actually covered over with cartilage in life. It's important in embryology, but that is well beyond the remit of what you need to know from medical school, so don't worry. It's just a giant hole in the center of the skull that's closed over. And if you actually look at any real skulls, you'll see this just sat there by the cavernous sinus with a sort of opaque looking bit of cartilage hanging over it. Sometimes that's even fallen off in the embalming process, but it's a closed structure, so you don't need to worry about it. This is a giant structure that's really easy to spot, and it's called the foramen ovale. And two of the main structures it transmits is V3, or VC, that should say. So VC, which is the mandibular branch of the trigeminal nerve. So it's the lower sensation to the face and it's the muscles of mastication. So remember masseter that we talked about there, but also temporalis and muscles within called the pterygoid muscles and a few others, but don't worry about that. And then we have this little structure here called the foramen spinosum. Now the foramen spinosum is really important because it transmits the middle meningeal artery. And the middle meningeal artery runs by a little structure here called the pterion. And a lot of people joke that the pterion is God's little joke because it's one of the thinnest portions of the skull, just on the side here, and one of the weakest portions of the skull, and a giant artery passes right by it. So if you get hit or fractured in that one specific place, you get a massive bleed on the brain. And then the final structure here is the internal acoustic meatus. Now, just before we mention this, you see this grey structure here that looks a bit craggy. It looks like a hill. Well, this is the petrous temporal bone, and petrous literally means hard, as in hard as a rock. And this is one of the most solid structures in the whole skull. And if you were to sort of like chip this open, you'd find the inner ear and all of the little vestibulocochlear apparatus and all those different structures, and then you'd find the middle ear. So you'd find all of the ear apparatus, but you'd also find cranial nerve 7 and cranial nerve 8. So they exit the skull via this internal acoustic meatus. So those are the key structures you need to know there. And then the posterior cranial fossa, there's three things. So here's our internal acoustic meatus and our petrous temporal bone. You remember this is the ramp, the clevus that we talked about before. And then you have this giant hole here called the jugular foramen. And this transmits three cranial nerves, cranial nerve 9, 10, and 11, as well as its namesake, the internal jugular vein, which drains the whole brain and a load of surrounding tissue and then drops out of the skull to go back to systemic circulation. The next structure here is this hypoglossal canal. And this is where cranial nerve 12, which moves the tongue, like this, moves the tongue. I didn't need to demonstrate that. That's how that exits the skull. And then the foramen magnum, the key things you need to know for the foramen magnum is that it transmits the brain stem and then into the spinal cord continuously, as well as the vertebral arteries that run up and then unite together on the clevus here. So one vertebral artery, two vertebral arteries, unite together on the clevus to form the basilar artery. And then finally, cranial nerve 11. So cranial nerve 11 actually begins in the lower cervical regions and then runs up through the foramen magnum and then pops over and out through the jugular foramen. So it starts out of the skull. So there's a lot of debate amongst boring anatomists as to whether cranial nerve 11 is even a cranial nerve, because it doesn't start in the cranium, it starts in the spinal cord. But, you know, whatever. So I hope this works. I tried to do this augmented reality thing the other day. So hopefully we can zip into the skull here. Yes. So just a quick review. So here's our crista galli here and the cribriform plate. We've got the optic canal here and the superior orbital fissure sat up there. You can see the foramen rotundum, ovale, and spinosum. And these three sort of are in a semilunar arrangement. So they're sometimes called the semilunar foramina, the foramen rotundum, ovale, and spinosum. And then you've got the foramen lacerum here, which is closed in life. you have the internal acoustic meatus here, and then this giant jugular foramen there. So these are the holes you absolutely need to know, and you need to know what's running through them. So you have the hypoglossal canal just here, cranial nerve 12, and then the giant foramen magnum. So that's my attempt at using augmented reality. So you should be able now to pick up a skull like David Tennant here, and you should be able to name all of these different structures. And now we're 20 minutes in, we're nearly halfway through. So what I'm going to do is I'm going to say pause, take a break here. If you don't want to take a break, if you want to keep on learning, I'm going to go and warm up my cup of tea because it's gone cold. There are now 9 multiple choice questions that are going to automatically play. So go make a cup of tea. We'll be back in 5 minutes. Otherwise, here are 9 multiple choice questions written in a similar style to the kind of thing that Tunstall deals with. And Tim Morris has just asked if there's an important artery that runs over the foramen lacerum. The answer is yes, and there's nerves as well. But for the most part, they're unnamed, and they're certainly not important to phase 1 at all, because the whole point about the arteries and veins is they're incredibly variable for their actual course. So in some people, they run over the foramen lacerum. In some people, they don't. So don't worry too much about that. So I will now give you 9 questions, or go and warm up your cup of tea, and I'll be back in 3 minutes. Long day in GP today, let me tell you, Christ. But it's good fun. Cool. Right. So before we move on to the brain, which we could talk about forever, but I'll try to limit to, I want this lecture to be done in an hour, so it will be over shortly, because I'm hungry and I haven't eaten yet, and you have enough to learn. Any questions? Anyone at all? Just text your questions and I will answer them at the end, or someone can unmute and ask me a question now if they want. Yes, Laura, I'm very proud of you. I'm very proud of you for getting the questions right. Right, then we will push on to part 3 of the brain. So, just a quick message. If there's anybody at all who is interested in brains, so psychiatry, neurosurgery, or neurology, we are... yeah, so if there's anyone who's interested in those things or surgery of any kind, there's this thing that Southampton host each year called the National Undergraduate Neuroanatomy Competition, which is a spotter of prosections of brains with little pins in them. And it says, what is this structure? What does it do? What happens if it goes wrong? And then a multiple choice question similar to the phase 1 exams that you have, but slightly harder. It's become the sort of premier thing to have on your CV if you're interested in anything psych, neuro, or neurosurgery, or any kind of surgery at all, because neuroanatomy is considered to be the hardest type of anatomy, and anatomy is one of the most respected things in the field of surgery. So if you're interested in anything like that, I would strongly recommend that you would consider doing that competition. And the NeuroSoc committee, of which I'm one of the members, we've made a curriculum that takes you from what is a cell in the brain to the level of neuroanatomy that you need to know for this competition and beyond. And they're all videos. They're all posted already. Probably about 80% are posted already. And actually, I'm going to talk you through the cerebral cortex lecture now, so you'll have some idea of what it's like. But if you've any interest in the brain or any interest in anatomy, just check out some of those videos and maybe join our team for next year. It's normally December, January when the competition is. But there you go. Let's begin. So, outline brain divisions as forebrain, midbrain, and hindbrain. So our brain has 3 different regions, if you were using the most gross simplistic thing. The major brain divisions are the forebrain, the midbrain, and the hindbrain, and they're named not so much for their location, but for when they evolved phylogenetically. So we began with a hindbrain, then a midbrain grew on it, and then a forebrain grew on that, basically. And a lot of species only just have hindbrain, still alive today. Some have a bit of midbrain, some have a bit of forebrain, but we have the full thing, basically. So the forebrain, or the prosencephalon, is divided into the telencephalon and the diencephalon. The midbrain is just divided into the midbrain, it's nice. But the hindbrain is also called the rhombencephalon because it looks rhomboid in shape in embryology, and it's divided into the metencephalon and the myelencephalon. What does this mean for you? Well, it's a little bit of embryology because Tunstall likes embryology, but the telencephalon ultimately becomes your cerebral cortex and the basal ganglia underneath. The diencephalon becomes the thalamus, the hypothalamus, and a few more things, but unless you want to go on a journey of the neuroanatomy syllabus, it's just the thalamus and the hypothalamus. And then the mesencephalon is just the midbrain, the top of the brainstem. The metencephalon is the cerebellum and the pons. And then the myelencephalon is the medulla oblongata. So these are different terms, but the overarching picture you need to know is that the midbrain is the middle, everything above it is forebrain, and everything below it is hindbrain. So your midbrain is that midpoint. So the cerebellum, pons, and medulla are all hindbrain structures, and everything else is forebrain. Okay, so in brief, you need to know, as a very minimum, the major lobes of the brain. So we begin here with a frontal lobe, then we have a parietal lobe, and they're named for the bones that protect them. So frontal lobe, parietal lobe, temporal lobe, and occipital lobe at the back. And if we look here, we can see It's not that detailed, so we'll go into a better illustration later. But a lot of people get that lateral view. They've memorized it, they're happy with it. But where they get you is the medial view, because this medial view is very rarely seen, because the lateral view is much simpler looking. So you need to be able to name these 12 structures on an open brain. So you're holding both hemispheres, you lift them apart, and then you look inside. Can you name these 12 things? So if this is a recording, pause and see what you can go through. But in the interest of time, I'm going to briefly talk you through them. So number 1 is the frontal lobe. Nice and straightforward. Number 2, this region here is called the cingulate gyrus. And a gyrus is a groove, as opposed to a sulcus, which is a hole. So you have 2 grooves, 2 gyri, with a sulcus in the middle. Okay? So this is a cingulate gyrus. And cingulum basically means the border. It's sort of like a limbus, if you will. So it's the cingulum around the corpus callosum. So it's a portion of the cerebral cortex that wraps around this structure called the corpus callosum, which is label number 11, which we'll get to. Number 3 is the continuation of the central sulcus. The central sulcus is huge laterally, but it's actually really difficult to spot medially here. But it's really important because that then tells you where the primary motor cortex and the primary somatosensory cortex is, which we'll get to later. Number 4 here is our parietal lobe. And number 5 here, well, here's our parietal lobe and here's our occipital lobe. So this is our parieto-occipital sulcus, a sulcus between the two. It's great when they're not named for people, they're named for where they are. Number 6 here, this is the occipital lobe. And then number 7 here, this is the calcarine sulcus. This is really important because if you open up the calcarine sulcus, this is where almost all of your visual processing centers are. It has its own calcarine artery, and this is the region where the center of your vision, if you look at anything in your room right now, anything, when you look, the center of your vision is crystal clear and your periphery is blurry. The center of your vision is being processed initially by the banks of the calcarine sulcus, it's called. So it has its own discrete blood supply because it's so important, because you cannot read, you cannot pick a berry off a tree as a caveman and look and see, is that a poisonous one or not, if you don't have crystal clear central vision? So the calcarine sulcus is a really important structure to know because it is responsible for your center of visual acuity. Now, number 8 here, this is our subparietal sulcus. So the parietal lobe begins here and it ends down here. So this is the subparietal sulcus. Number 9 here, this is the last main lobe remaining. It's the temporal lobe. And then number 10 here, this is less important. So number 10 and 11, you've got the corpus callosum here, which is a huge, thick, tract bundle of white matter that connects both hemispheres of the brain together. So you can talk to each other bilaterally and you can have some cooperation between the two hemispheres. That's the corpus callosum's job. So above it is a sulcus. So there's a space between the corpus callosum and the cingulate gyrus, and it's imaginatively called the callosal sulcus, which means that 12, because it's the space between the frontal lobe and the cingulate gyrus, is imaginatively called the cingulate sulcus. So this medial structure set are the things that you need to know, okay? Because everyone knows the lateral, so they never show you the lateral and have you label it. They always show you the medial. So the diencephalon, two of the brain, encephalon, is the thalamus and the hypothalamus. And it's this deep structure in red here. It's this deep central structure. It's the thalamus, 2 egg-shaped clusters of nuclei. The thalamus used to be called the gates to the cerebral cortex, i.e., if you're thinking of anything, it has to pass through the thalamus and project to the higher cerebral cortex forebrain in order for you to be aware of it. All of the other stuff that is sort of subconscious, that you're not really aware of, that you're in therapy for, that doesn't go through the thalamus. And I'll give you an example. So now everybody feel the sensation of your tongue in your mouth. And now everyone can feel a tongue in the mouth and you think, oh. Now if you do what's called electroencephalograms and you put electrodes on the nerves of your tongue into the brainstem, you see there's no change. It's always continuously firing sensory information, and it comes into your brainstem and up to your thalamus, and your thalamus makes a decision through processes we don't understand to say, it's not time for Dan to be thinking about his tongue. So until something happens in the environment that means I need to pay attention to my tongue, my thalamus keeps batting away that information and pushing it down into the hindbrain and the midbrain and saying, he doesn't need to know, he doesn't need to know. But as soon as it's relevant, so now I've mentioned it, everyone's thinking about their tongue in their mouth, that same information that was always there and ready is allowed. So the gates of the thalamus open. So that's a really important thing. Nothing major happens in the brain without the permission of the thalamus. Below it is the hypothalamus. Hypo literally meaning an inferior structure. It's below to the structure. So here's our thalamus, here's our hypothalamus. And the hypothalamus is said to be the conductor of your endocrine system. And your endocrine system is how the body responds to everything going right and wrong inside of it and everything going right and wrong outside of it. So it's the conductor of your day-to-day life, basically. So these are two vitally important structures, and any damage to these structures are going to have profound effects on patients. So the diencephalon, these twins of thalamus and hypothalamus, you absolutely need to be aware of. So the other thing you need to be aware of is the basal ganglia, or the more correct term is actually the basal nuclei, because they're a cluster of cell bodies in the central nervous system, which makes them a nucleus, not a ganglion, but basal ganglia sort of caught on, so we'll use basal ganglia. Now, I'm not going to go through these structures, but this is the classic cross-section that you need to know. You need to know where the caudate is. You need to know where the putamen is. You need to know where the globus pallidus is. So here's your caudate, here's your putamen, here's your globus pallidus. And this is moving not into anatomy, but into neuroscience and into the basal ganglia lecture, which I think you're going to have later, or it's in the movement and the tracts lecture. They're going to talk about the different direct and indirect pathways and what's going on here. But for now, you need to appreciate the structures, because there could easily be a question where there is this picture or a prosection picture with a label. What is this structure? What does it do? Kind of thing. What is its blood supply? So caudate, putamen, globus pallidus, and then here is our giant thalamus, which means the nucleus below it is the subthalamic nucleus. And then the one below that, so here's the beginning of the midbrain, it's the substantia nigra. And everyone knows the substantia nigra is about Parkinson's disease. So if you see any stem with substantia nigra, it's Parkinson's disease. So you need to be, everyone knows the cerebral cortex and the frontal, parietal, temporal, and occipital lobes, but you need to also pull off that cerebral cortex and make sure that you understand what this weird alien thing is here. So here's our caudate. Here's what it doesn't look like in that's cross section, but here's what it looks like side on. Caudate, literally meaning tail, a cord, is a tail. It wraps around in this orange way here, from the front right the way to the back. Then medial to that is the putamen, and medial to that is, well, the thalamus and things, but we can't really see them here. Don't forget a few other structures like the ventricular system, which is not the remit of this lecture, but you need to know the different ventricular systems and how CSF flows through them and their relation, because when you have a blockage of CSF and the ventricles start to bulge, they will bulge into the caudate nucleus or the putamen, or they'll bulge down into the amygdala, and they'll cause very stereotyped problems amongst the patients. So here's that weird alien picture as a 3D model. As you can tell, I like my 3D models. The green structure centrally here is actually the corpus callosum. The corpus callosum is there, and then this horrible sort of like I don't even know what color you'd call this. This is the brainstem here. And then the yellow structure here is actually the hippocampus. So that's the hippocampus. And then the blue structure here is the caudate and the putamen. So try to look at 3D structures like this. It's really hard because in the anatomy lab, they have prosections of these that you can look at and turn around, but it is really important to be able to visualize these structures here and their relations. So in the median plane, there's the corpus callosum, and then you've got your caudate, and then you've got your putamen, and all those different kinds of things. So you'll be pleased to know that was the largest learning outcome, and we're powering through now. So we're going to talk about the key clinical correlates of gross lesions to the major portions of the brain. So if we begin with the frontal lobe, the frontal lobe here is divided into several areas, but the most important ones for it is damaged and the patient in front of me is therefore exhibiting these signs are these three regions: your motor cortex, Broca's area, and the prefrontal cortex. If I pause this, if you find the central sulcus, then immediately anterior to the central sulcus is the primary motor cortex. And this is where we introduce the idea of primary cortices and association cortices. Primary cortices are dumb. They're really dumb. They don't know anything. They execute commands. They do as they're told, okay? So the primary motor cortex knows nothing, but just does as it's told. So if you want to move your hand, the primary motor cortex lights up just before you move, but it doesn't know to move the hand unless the plan of moving the hand has been passed to it from another area of the cortex. So the primary motor cortex will execute the command from the association cortices. So the association cortices are the brains of the brain, if you will. That's where most of the planning goes. So here's our primary motor cortex. And then this huge swathe of tissue in front of it here is the association motor cortex. And this region here is called the premotor cortex. And the premotor cortex comes up with complex plans for movement. And these regions are on fire in scans for piano players and people that do complex movements because they've developed very fine plans. Then you have what's called the prefrontal cortex. So we have our primary motor cortex, we have our premotor cortex at the front. Ooh, what's happened there? Premotor cortex at the front, and then, what's happened there? Sorry about that. Yeah, so we have our we'll spin it back around. So primary motor cortex, and then we have the premotor cortex here, and then we have the prefrontal cortex here. Now the prefrontal cortex, this is about emotional regulation and intellect. In short, when you're drunk, the prefrontal cortex is turned off. So it's all about learning the social cues of the environment and being able to self-regulate. And I will leave you to decide who has a well-developed prefrontal cortex and who does not. So the parietal lobe now. The parietal lobe, the wall lobe, the most important thing here is that you remember the concepts of primary and association, because the same is true. Primary executes the commands and association is where all of the ideas come from. So here is that central sulcus, which means this is the primary motor cortex again. But if we go posterior, we find the primary somatosensory cortex, which is this region here. Okay, this receives all of the information in the brain and in the body. It receives all of the sensory information. It doesn't know what to do with it, but it passes it to the association cortices throughout the body for them to figure out what it's seeing. Okay, so when I'm tapping this table and I can feel the wood surface, that wood surface feeling is transmitting to my primary somatosensory cortex, but the primary somatosensory cortex is saying, okay, this feeling's appeared, I don't know what it is, I'm going to send it to an association cortex because it can translate, it can tell me, okay? The rest of the parietal lobe, that isn't this strip here, the primary somatosensory cortex, the rest of it is the parietal association cortex. This is a huge thing for figuring out what is coming into the brain from the outside world. This is the dictionary that looks up all of the different things that are appearing based on your memory and your experience, all those different things. So if you have lesions to it, guess what happens? If it's the primary somatosensory cortex, you aren't aware that you're feeling things at all. The nerves are still experiencing the phenomena and trying to transmit it to the brain. The thalamus might even say, hey, I've noticed a burning sensation in the left foot. That sounds like it's important. I'll throw it into the primary somatosensory cortex so Dan's aware that his foot's on fire. So you throw it in, but the primary somatosensory cortex had a stroke. It's damaged, so it doesn't know. It's not able to receive that information, and then the dictionary, the parietal association cortex, isn't then able to look it up. So if you have a primary somatosensory cortex damage, you have contralateral hemianesthesia, because the left side of the brain looks after the right side of the body and vice versa. So contralateral one-sided anesthesia, loss of sensation. But if it's the parietal association cortex, you will have problems with the dictionary, problems with understanding what's going on. And one of the easiest ways to test this is to get your keys and to have the patient close their eyes and put a pair of keys or something very familiar like a tennis ball in their hand, close their eyes. What is this? Because what you're doing, think about it, is they're feeling the sensation. It's going from the hand into the spinal cord, up into the brainstem. The thalamus is saying, someone is asking me what's in my hand, so it's important. So I'm going to open the gates and allow it to enter my cerebral cortex so he can think about it. So what does the thalamus pass it to? The primary somatosensory cortex. So it says, hey, I've got this sensation in the hand. I need to send it to the dictionary to look it up. But the dictionary is damaged because there's been a stroke to it. So it has astereognosis. Stereognosis is the ability to know what things are based on feeling for them. Like when the room is dark and you can feel for a light switch. If you can't do that, the parietal association cortex has suffered a very specific stroke. So part of an advanced neurological exam is having a patient close their eyes and pick up a very familiar object. What is this? And you're testing the parietal association cortex. So the temporal lobe. The temporal lobe, dead easy. It's this side lobe here, and for the most part, it's involved in, sorry, I'm being called. This is hearing. And how do you remember the temporal lobe is hearing? Because it's right by the ears, right? So your auditory cortex is in here. It's by the lateral fissure. It's sat in there. And it is super important for hearing and for processing the hearing information, okay? If you were to take that temporal lobe that I can see there and peel it off, attached to the back of that temporal lobe, deep inside, you would find the hippocampus and the amygdala. And for all extents and purposes, you need to know that the hippocampus does two things and the amygdala does one thing. They both do about 50 things. But for all extents and purposes, unless you want to be a neurologist or a neurosurgeon, you only need to know these things. The hippocampus does memory, mostly short-term to long-term memory, but the hippocampus does memory, as well as these things called place cells, which won the Nobel Prize in 2013. And that enables you to have a sense of direction, basically, to remember your way to the shops, to remember your drive to work. That's the hippocampus. And the amygdala, fear and emotion, anxiety. Just think about that. Obviously, there's more stuff. But if it's this superficial temporal lobe, it's hearing, it's auditory sensation. If you pull that off, you find the hippocampus and the amygdala. And those structures, memory, place cells, fear and emotion. And then finally, we come to the occipital lobe, the easiest of them all. It's all vision. We've already spoken about it. So you will have visual field defects. And these are very weird visual field defects because the information is fine, the eyes are fine, but the brain can't interpret the information. So they come out with some very weird and wonderful visual field defects, which you can categorize with ophthalmology. But for the most part, has the patient had what you think is a stroke? Yes. Is their vision impaired? Yes. Okay, I think there might be a bleed or ischemia to the back of the brain, so I need to scan the brain. That's basically to the level at which you need to know it, unless you're going into these things more specifically. And there's going to be a lecture on the visual pathway, so you'll do all the homonymous hemianopias and all the visual field scotomas and defects. We won't focus on that now because there's so much to cover in skull and brain, right? So this is lifted from a book, which is basically, I can link it to you if you want it later. It's basically everything a medical student in F1 needs to know about the basic science to understand clinical medicine. And this is what they say you need to know about the cerebral cortex ever, unless you're going into the specialty. You need to know there is a central sulcus, and in front of it is the primary motor, behind it is the primary somatosensory. In front of that is the premotor cortex, which is the important one. It knows what it's talking about. And then behind it is the somatosensory association cortex. These are the dictionaries that work out the dumb stuff that's in the primary regions. You also need to know about this thing called the frontal eye field. This helps control movements and nystagmuses, jumping, jerky movements of the eyes, which will be covered in the cranial nerves lecture. So don't worry about that for now. And then the prefrontal cortex, the thing that's turned off when you're drunk. That's what you need to remember. There is this area of the frontal lobe that we briefly touched on called Broca's area. And then at the back, here, at the top of the temporal lobe, is Wernicke's area. So Broca's and Wernicke's. Broca's area is about expressing language. So it contains all of the motor commands to move the vocal cords and your mouth to phonate, to pull air from your lungs and to turn it through your anatomy in such a way that you can speak. It's involved in that process, expression. But Wernicke's area is about the understanding. And this makes sense, right? Because Wernicke's area is in the temporal lobe, right by the auditory cortex, where sound information comes in and is processed. So it makes sense that the neighbor of the primary auditory cortex would be Wernicke's area, which is about the reception, the interpretation, the understanding of language. They are connected by a white matter tract called the arcuate fasciculus. And this is really important because if this is cut, the patient can hear what is being said because Wernicke's area is fine, but it can't then reply to you. And this is called a conduction aphasia. Aphasia, A meaning non, phasia, speech, right? So a conduction aphasia is when this is cut because I can understand what you're saying, but I can't reply to you. It's really quite sad if you think about it. And it's pretty similar to Broca's, right? But when Broca's is damaged, there'll at least be an attempt to speak. There'll be some, it will sound very strange, but they'll be attempting to speak. If the arcuate fasciculus is cut, it's much less likely that there will be an attempt to speak. So I hope that makes sense. And then all that's left here is the primary visual cortex and the association cortex. So the primary visual takes in what you're seeing and sends it to the the visual association cortex, which is the dictionary, which looks up everything you're seeing. Oh yeah, I know what that looks like. That looks like a tennis ball, which I learned about when I was 2. So here's a real brain. The sulci and gyri that we've talked about, I'm going to briefly mention them again because repeat, right? So here's the corpus callosum, and you need to know the callosal sulcus here. Here's the cingulate gyrus, and you need to know the cingulate sulcus along here. Here, this is the parieto-occipital sulcus. And here's the all-important calcarine sulcus. If I peel apart that calcarine sulcus, I find V1 at the pole here, right at the back, the primary visual cortex. Those are the main ones you need to know. And then on the lateral side, you need to know the central sulcus. Here's your giant central sulcus here, which means that your primary motor cortex is here and your association motor cortex is here. And then behind it, there is the primary somatosensory cortex and the association cortex behind it. And then just bear in mind this little fissure here called the lateral fissure, which is named after a guy called Sylvius. So it's the lateral Sylvian fissure, but nowadays just call it the lateral fissure because names of long dead people, no, just the lateral fissure is fine. And if you open that, that's where the primary auditory cortex is. So know where the primary motor, the primary somatosensory, the primary visual, and the primary auditory is. Here's Wernicke's and here's Broca's. Don't worry about knowing what the exact wiggly bit is for Broca's, because brains are very variable. If you dissect 10 brains in a row, there's a lot of permutations between them. So you need the approximate geography. There's a lot of overlap anyway. It's a bit like the dermatomes of the body. There's a lot of overlap. So just get a rough approximation of where these are. So this is something that I'm not going to cover now, but there's a recording here, and I strongly advise you to go back and see if you can think about what we've just gone through, look at all of these numbers, and then label it. So pause the video now, and then here are the answers. So this is an actual brain, and let's see if you can take that information and apply it to a real brain. And then what else do you need to be able to do? So here's another example. So see if you can do it on a second brain, and then see if you can do it on an all-important scan. So most doctors will spend most of their life looking at the brain, not as a neurosurgeon does, with a hole in a skull bleeding everywhere, but via a scan. So more important than being able to pick it out on a drawing or pick it out on a prosection is, can you pick it out on a scan? The corpus callosum here looks gorgeous. There's the corpus callosum. Here's our thalamus, here's our hypothalamus below, and this little hole here, this is the sphenoid bone, and this is the sella turcica. This is where the pituitary gland sits. So see if you can pause the video here and go through all of those numbers. Where are they on this brain? And compare them to actual brain. And if you can do those two, you've got it. You've got it made. You've absolutely nailed it. So, organization of the motor and sensory homunculus. I'm going to keep this really easy, okay? Really easy. Wait, I don't know why that's like that. Why is it not that? It skipped a thing. Interesting. Okay, so never mind. The gross anatomy of the medial sagittal view. So you need to be able to interpret this view on a CT and an MRI. You need to be able to look at those things. So questions like, where is the corpus callosum? What is the name of the area dividing the cingulate gyrus from the corpus callosum, which is something we've covered? Can you find the parieto-occipital sulcus on there? Can you find the pituitary gland on there? Can you name all of the lobes on there? And can you spot the thalamus and the hypothalamus? Can you name the different regions of the brainstem? And can you spot the cerebellum? That's the level that you need to know. And then there'll be a picture like this. What is the yellow arrow pointing to? And it's the lateral fissure. What cortical region lies deeply within the yellow arrow's groove? It's the primary auditory cortex. And if a patient has an expressive aphasia, where is the lesion based? So if they can't speak, where is that lesion? And then which artery leaves the sulcus labeled? So it's really important that when you draw that circle of Willis, that you know where the anterior, posterior, and middle cerebral arteries go. In this case, the middle cerebral artery pops out of this lateral fissure and supplies superior branches and inferior branches. So it has a load of supply here that we'll come to in a minute. So, sorry, we'll get to the homunculus now. So we have said that this is the central sulcus. And anterior to it is the primary motor cortex, and posterior to it is the primary somatosensory cortex. Now, neuroscientists don't like saying primary somatosensory cortex, so they call it S for sensory 1, because it's primary, and primary motor, M1. So there's M1 and there's S1, and just if you want to, you can actually call it M1 and S1 because it's well-recognized nomenclature. Now, we've said that the association motor cortex is called the premotor area. It's about planning movement. But on the medial portion, so here's the medial brain, right? Corpus callosum here, so the cingulate gyrus is there. And if you push up a bit, you can see that this is M1. This is a tiny area. So the primary motor cortex is very little space medially, very little. The majority of it is made up with the rest of M2, or the motor association cortex. Medially, it's called the supplementary motor area. And the supplementary motor area is the starting pistil. So the premotor area laterally comes up with the plan, premotor plan, supplementary start. So there's alliteration so you can remember it. The premotor laterally is about planning and the supplementary motor is about starting it. So here's the way that information flow will go. I want to pick up my cup of tea. So my premotor cortex comes up with the plan by which my hand can grasp this cup. But it can't do anything until the premotor cortex gets a permission slip from the supplementary motor area. So we start out here and we fire fibers medially to the supplementary motor area. And that says, yeah, okay, I'm thirsty. You can do it. So it allows it to start. The firing pistol goes. So we have the plan and we have permission. So now all that's left to do is to execute on the motor command. So we start for any movement with premotor, then we go supplementary motor, then we go primary motor. And that's the order of flow. Obviously, it's considerably more complex than this, and there's a lot of crosstalk between them, but for the most part, that's the way it goes. This is the homunculus. And if I were to describe the homunculus in a single sentence, it would be, imagine that the bits of the body were a size proportional to how much of cerebral cortex it gets. So this map, this homunculus, shows you at first glance which areas of the body your cortex prioritizes. And what a surprise for human evolution, which thrived on tools, that the hands are huge. And what a surprise for human evolution that thrived on speech and language, that the mouth and the tongue, both essential to speech, is huge. So this is the primary motor cortex, and this is the way it's arranged. So here is the right slap bang middle of the brain, and here's the lateral aspect of the brain. To orient you, this is the lateral fissure. So here's a bit of parietal lobe, and here's the temporal lobe. And then popping out of here will be the middle cerebral artery, which we mentioned before. Now, what you need to think about for the homunculus is, imagine you're sat. You're a guy, right? And you're sat on the top of the brain and your feet dangle in between the two hemispheres. So there's your butt and your feet and legs dangle between those two hemispheres, and then you lie backwards. And you're good at yoga, unlike me. So you lie backwards and your whole body contorts backwards. But what that means is that this central medial portion is the lower limb and your butt. The middle bit is your torso, and then your arms and your hands, which lie out backwards. And then it's your face, and then it's swallowing. Why is this important? Because you can localize where a bleed or where a clot is based on how a patient presents. If a patient can't lift their arms up, you can pretty much guarantee that a lesion may well be here, right? So knowing this topical organization of the homunculus is really important. So here's the circle of Willis. Just think about the big picture right now. We have anterior cerebral arteries that run in the middle, and we have middle cerebral arteries that run to the side, which means that the side of the homunculus is supplied by the middle cerebral artery. and the middle of the homunculus is supplied by the anterior cerebral artery. So when a patient has a stroke in the anterior cerebral artery, their upper limb and their torso is spared, it's safe. But their lower limb, their butt, and their bladder and bowel is not spared. So they can't move their lower limbs, they're paralyzed, and they're incontinent. So if you have that presentation, you're thinking there's a stroke in the anterior cerebral artery. So now briefly, gross blood supply. So we've already briefly touched on this. So here is a lateral view. You can see the lateral sylvian fissure. The middle cerebral artery pops out sideways, pops out of the lateral fissure and supplies a superior branch and an inferior branch. If we turn the brain medially, most of this is anterior cerebral artery, because the anterior cerebral artery, the circle of Willis is around here, and the anterior cerebral artery moves, you guessed it, anteriorly to supply this region, and then it loops over the corpus callosum like that. So it has an arcade that loops around. So it supplies this looping structure medially, right? And then the bottom of the brain and the back of the brain is the posterior cerebral artery. So what a surprise that anterior cerebral artery is the front, posterior is the back, and middle is the side. Okay, so that is in a nutshell what you need to know about the cortical distribution of this blood supply. What does that look like for the homunculus? As we said, anterior cerebral is the lower limb, the hips, and coming up to a little bit of the trunk, and then everything else is middle cerebral. So if a patient can't lift their arms above their head, is having trouble speaking, but they can tap dance, then you know that the middle cerebral artery is stroking. And note that the motor homunculus is not touched by posterior cerebral artery. So you can also say he's got predominantly motor symptoms, this guy who's having a stroke, so I don't think it's the posterior cerebral artery that's damaged. And this is the thing. Print this off, put it on your wall, learn it in your sleep. You need to know the circle of Willis. You need to know the circle of Willis. So go back to this video and pause it, because you need to know the circle of Willis. Did I mention you need to know the circle of Willis? I don't know if you have short answer questions anymore, but a few years ago, the Tunstall question was draw the circle of Willis and label it. So that is the kind of thing they're expecting. If it's MCQs predominantly, then the kind of thing he's going to say to you is, here is a pin. What is this artery? Which of the following structures that I'm about to list does this supply? So you need to know that. That's really important, really important. You have a stroke lecture, I think, in phase 1. Make sure that you look at the different territories of these arteries and make sure that you can pull that out. Very important. In a nutshell, the middle cerebral artery going sideways causes sensory and motor loss to the face and the upper limb. Okay? Middle cerebral artery is the face and the upper limb. The anterior cerebral artery is the lower limb and incontinence. Okay? So that's the most important thing to know. But also, because the middle cerebral artery is doing the lateral stuff, if it's in your dominant lobe or your fluent lobe, the lobe that's responsible for language, because language is lateralized to one side of the brain only, not both sides, if you have a stroke in your language side of the brain, you're going to have an aphasia. And it might be Broca's, it might be Wernicke's, it might be that arcuate fasciculus connecting the two, it might be a conduction aphasia. If it's in the non-dominant hemisphere, you get what's called hemispatial neglect, which is very similar to that astereognosis. You neglect one side of the body. And the classic presentation of this is, it's so bizarre because it's an unknown unknown. You don't even know that you don't even know that you have two sides of your body. So the patient will present to you with a half-shaven face, and they will look at themselves in the mirror and completely hallucinate out the other side of themselves and imagine they have a beautifully shaped face. they'll have only put their coat on in one arm. It's very apparent, and the way that they walk will be staggered in one way. So the dominant versus the non-dominant. So that's what you're looking for. A posterior circulation, it's the back of the brain, right? It's the back of the brain. So it's this concept of hemianopias. Hemianopias, half, and then opsia is vision. So half and is absence. Half absence of vision. So because of the way that the visual field and the visual pathway moves, half of the visual field goes to one half and half goes to the other half. So half of your visual field will be cut off, called a scotoma, which you'll do in the visual field lecture. If the dominant lobe is affected, you get something called alexia sans agraphia, or alexia without agraphia. And this is the last thing I want to mention today. And it's one of those things that it's sort of like the top kind of students are thinking of. Alexia without agraphia. Lexia, like a lexicon, is to read. So alexia means cannot read. So you cannot read without you cannot write, which is a weird double negative or triple negative kind of thing. So it means you can't read, but you can write. So if you closed your eyes and I said to you, write your name, you could write your name. And then if I told you to pick up that piece of paper and read out what you just wrote, without your memory of knowing what you wrote, you would look at it and it would look like hieroglyphs. It looks bizarre because your brain receives the information to the primary visual cortex, but look, the dictionary is cut. You can't look it up. So because you can't look it up, despite having perfect vision, it just looks like gibberish. So even though you've learned to read, it just looks like gibberish. So you can write, but you can't read. And have a think about why you could write, but you couldn't read, which is quite an interesting concept. So that is that. There's a couple of extra questions here which are answered on the slides, which I'm going to leave up for the video because you've gone through a lot, a lot. What I want to briefly mention is the website that we've made. So because of COVID, we've thought we need to put peer support virtual. So there's this website that we have called MedGuide, which you might have heard of. There's 1,400 questions for phase 1, all MCQs. Every question has to be about a phase 1 learning outcome. And it's actually exploded around the world more than it has in Warwick, which is strange because it's tailored to the Warwick curriculum. 7,000 medical students around the world have now used MedGuide since the start of 2020, which blows my mind, but very few Warwick medical students. And we have loads of questions, loads of questions. We've got 30 volunteers and every single one has passed really well at phase 1. So what I'm saying is use MedGuide. It's medguide.uk. Use it and we want you. So I know you've got an exam coming up, but when you pass this exam, which you all will because you care enough to be here watching Teams at 7 o'clock at night instead of eating, and I'm about to eat my weight in cookie dough, we want you, right? So email me if you want to write some questions for MedGuide, because more than a master's degree, doing peer support gives you more points. Teaching medical education gives you more points on junior doctors' portfolios, so you're more likely to land the job that you want going forward. And all you need to do is link to the page of questions that you've written. It's super easy. We have a good editorial process to make sure that what you're doing is right. So save my email and email me when the exams are done, or email me now and just say, hey, I'm interested in doing this in the future, because it's good for your CV and it makes our job easier because we can just get more volunteers. So we're done, basically. Please do scan this QR code and it will take you to a super, super quick feedback form that you can just fill out nice and quick, because the feedback form goes on my CV. So that's the thing that I get for the unpaid lectureship here. And don't forget MedGuide. So I'm going to leave this up and we're all done. So, yep, enjoy your night and goodbye. Bye. How do I stop the recording?